this podcast, we're going to look at the 4 different levels of protein structure, starting with a single chain of amino acids all the way up to the 3-dimensional structure of quaternary proteins. All right, so we always start at the primary level of proteins for protein structure, and the primary protein structure is the order of amino acids that are joined together as a polypeptide. That is our primary protein structure. So the sequence of those amino acids. And the sequence of these amino acids is very important because the R groups of your amino acids are all going to interact with each other. And the way they interact is going to dictate how that protein folds, and the shape of the protein is going to play a big role in the function of the protein. So primary structure is really important. Again, from the last podcast, we talked about how to look at a chain, a polypeptide chain, to figure out what amino acids are in there. So we just kind of draw our line at the amide bond between the carbonyl and the nitrogen. We can see that this has, and they're doing R1 instead of doing an actual R group for an amino acid. There's 1, 2, 3, 4 different amino acids in this chain. These little squiggly lines just tell us this is a polymer and this chain would continue on. So the primary structure is simply the order of amino acids. If we were to change the order of amino acids, we would change the shape of our protein subsequently. All right, in our secondary structure of proteins, we're going to start to get a 3D arrangement, and you're going to get this in localized regions of your protein. This is due to hydrogen bonding, and it's especially due to bonding between the carbonyl of one amino acid and a nitrogen group of another. So you have hydrogen bonding occurring. There are 2 types of secondary protein structure. There are more types, but these tend to be the most prevalent. The first is called an alpha helix. So what happens in an alpha helix is your protein chain, which was straight, starts to turn and coil in a clockwise spiral. So it kind of resembles a coiled spring. An example of this would be alpha keratins in your hair. And something to note, we're seeing an image of this right now, is you have hydrogen bonding between the carbonyl of one amino acid, which has a slight negative charge, and the nitrogen group of another, which has a slight positive, the hydrogen attached to that nitrogen. So you're getting some hydrogen bonding between different amino acids, and that's what causes it to turn up on itself and to bring parts of that straight chain closer together, and that's what gives us that shape. Your R groups of your alpha helix are going to extend outwards. Okay, so they're extending away from the core of the helix. The second type of secondary protein structure that we see most common is a beta-pleated sheet. These are where you have several protein chains held together in a parallel fashion. So we have your carbonyl. Let's see if we can find a carbonyl and a nitrogen group from 2 different amino acids. We're still seeing hydrogen bonding in these, but the way that they're arranging themselves is a little bit different. Your R groups can be oriented both above the plane and below the plane, which is what we're seeing in this image. Beta-pleated sheets tend to be a little bit stronger, whereas alpha helices, you're going to see that protein structure where you need flexibility because that protein has a little give in a coil shape, and we'll look at an example of this in just a moment. Beta-pleated sheets are favored by amino acids with smaller R groups. When you get larger R groups, you have steric interactions that are preventing those parallel strands from coming too close together. So therefore, it can't be stabilized by hydrogen bonding alone. All right, so something to note before we go into our first example is how we can show in a single protein the alpha helices and the beta-pleated sheets. We're going to use a shorthand diagram for that, and this is preferred, where your alpha helix is going to look like a green coil, beta sheets look like a purple arrow, and then of course you have some random regions. There are 3 different ways that we can show proteins, and those are shown below. There's a ball and stick model. And just looking at this, it makes me a little anxious because it's really hard to pick out the R groups and where the bonds are and what's going on. space filling model we will see more of later in this chapter when we talk about enzymes. We can get a good feel for an active site of an enzyme and the shape of the protein from a space filling standpoint. A ribbon diagram is probably the cleanest way to look at that. So we can see how many alpha helices there are in that protein, how many beta pleated sheets you have, and that gives us a good idea also of what the protein structure is. The protein that you're looking at down below is a lysozyme, and that catalyzes the hydrolysis of bonds in bacterial cell walls. And if we were to weaken them with a lysozyme, it would cause them to burst. So lysozymes can be useful in killing bacteria. All right, spider dragline silk is one of my favorite examples of how useful and how different an alpha helix region versus a beta pleated sheet region is. So what you're looking at here are pretty orderly repeated regions for this protein that's found in silk. And it is the beta pleated sheets that make spider silk so strong. They're parallel to each other. It provides strength. They're held together. However, it is these regions of alpha helices that make it elastic as well. So it has a little stretch to it because the coil can give and uncoil a little bit. So spider silk is exceptionally useful. It's something that we've studied trying to recreate because it could have a lot of uses. It's something that is incredibly strong, yet has that elastic property, so it could be useful in medicine. Say if we were to make stitches that could be both strong and elastic, that would be better than just having a piece of plastic that doesn't have as much give to it. In tertiary structure, this is also a 3D arrangement of your proteins. So we took a single amino acid strand and we allowed it to either coil into alpha helices or beta pleated sheets. And those beta pleated sheets and alpha helices can also further fold together and interact with each other. So when we talk about tertiary protein structure, we're talking about the bending and folding that results from a bunch of different types of reactions between the R groups of the amino acids in the protein chain. So these interactions can be hydrophobic and hydrophilic. Oftentimes with a protein, if you're in an aqueous environment like a cell, which is composed mostly of water, you're going to want to fold in a way that all of your polar groups are facing outwards and your nonpolar groups are facing inwards so that this protein can interact with water. If it was hydrophobic, it wouldn't be able to do that. So we see some folding as a result of turning those hydrophobic nonpolar portions in and the hydrophilic polar portions out. There are interactions between the hydrophobic or nonpolar regions within that protein that are of note too. So besides that orientation, we're also going to have London dispersion forces. This is going to be between two nonpolar R groups. We'll also see hydrogen bonding between R groups. So as we talked about extensively before, you're going to have carbonyls of one amino acid interacting with the amino group of another to form those hydrogen bonds. And those hydrogen bonds are also going to pull different portions of that protein chain together. So this blue ribbon represents the long extended primary structure, and we can see how those forces are pulling different parts of the chain together, twisting and turning it into its tertiary structure. The other thing that we have going on here that we haven't talked a lot about are disulfide bonds. Disulfide bonds are the result of an interaction between two cysteine residues. So here we have two cysteines that were oxidized. And when they're oxidized, they form a disulfide bridge or a bond, however you want to refer to that. That's pretty strong. That's going to pull this portion of the protein together with this portion and going to help it further fold. We also have some salt bridges in there. Salt bridges are ionic bonds between side groups. So if you have a basic amino acid and an acidic amino acid, those are going to have charges to them. Those full charges are going to be attracted, if they're opposite, are going to be attracted to each other. So we see that, and they're calling it in your book an electrostatic attraction. We have a positively charged basic R group, and we have a negatively charged acidic R group. So this difference in charges is going to create an ionic bond within our protein that is going to be a very strong bond, pulling two portions of that protein together and causing it to fold into a particular shape. In our final protein structure, quaternary protein structure, you have two or more folded polypeptide chains coming together as one protein. So in tertiary structure, we have our long chain of amino acids that's able to fold and twist and go through all the interactions that we talked about. If we were to get multiple subunits of that tertiary structure together, for example here, we have what's considered quaternary structures. So we have interactions between two or more folded polypeptide chains. We would consider that to be a quaternary structure. And just for note, I really like this image from your book because it takes us from primary structure, which is your individual chain of amino acids, to secondary structure where we see portions of the chain either going into a beta pleated sheet or an alpha helix up to a 3D shape in a tertiary structure, and then how those tertiary 3D shapes interact with each other. So I think this is a great rundown of all four levels of protein structure. All right, so here's a look at a quaternary structure of a protein with hemoglobin and myoglobin. Both of these proteins are considered to be conjugated proteins because they have a protein portion and a non-protein portion of them. Both of these share the same non-protein portion, which is the heme. That is a charged iron, Fe2+, that's surrounded by a large nitrogen-containing ring system. And the function of that iron is to be able to bind oxygen to it. So the function of each of these is a little bit different. We know hemoglobin is in the blood, and its function is to carry oxygen that has bound to the iron ion all throughout the body, delivering oxygen where it needs to go. Myoglobin stores oxygen in muscles and in other tissues. So it is actually the myoglobin that gives cardiac muscle its characteristic red color. Something else to note, looking at these two structures, is what happens when carbon monoxide is present inside the body. As we know, carbon monoxide will kill people, but it's due to the interaction of the carbon monoxide with the heme molecule. So what happens is, typically your heme and your myoglobin are going to bind oxygen and carry that around the body or store it for the body. When carbon monoxide is present, it is 200 times stronger in binding to the heme. Because of this, if a hemoglobin were to pick up carbon monoxide, it is not able to drop off that carbon monoxide. That carbon monoxide becomes stuck to the heme molecule in hemoglobin. Therefore, you're blocking oxygen from binding. Therefore, all of your cells are going to die because they're not getting any oxygen delivered to it. So think of carbon monoxide as being a molecule that's able to irreversibly bind to hemoglobin, and therefore that is why it kills people.